So Learning Unit 5 is on photosynthesis, and coupled with Learning Unit 4 on electron transport and oxidative phosphorylation kind of comprises the content that we're going to have for our third exam. And part of the reason I have these two learning units together for a test is when we learn about electron transport and oxidative phosphorylation in mitochondria, it's very similar to what we see in electron transport in photophosphorylation in plants. So again, we have these two organelles, mitochondria and chloroplasts, that are both in theory bacterial in origin. And so we're going to see a lot of homology between the kinds of things that they can do, but some very unique and different ways that they're able to do that. So we started out the chapter on photosynthesis really doing a comparison and a contrasting between some things that we've learned already in electron transport and oxidative phosphorylation in mitochondria, and then how things are going to be similar, different, complementary in what we see with plants in their chloroplasts. So some big picture review concepts as we think about sort of the main equation that we've spent the bulk of our time in biochemistry discussing, fuel metabolism or fuel oxidation. We see fuel kind of irreversibly being oxidized to carbon dioxide, water, and then energy. Again, the oxidation is the loss of electrons. And so those electrons end up being given to NAD plus and FAD. In their reduced form, they're going to deliver electrons to the electron transport chain. They again then become oxidized so that they can be reused. Loss of electrons is what we generate carbon dioxide. We see that in the pyruvate dehydrogenase reaction and then citric acid cycle chemistry. Electron transport in the mitochondrial generates our proton gradient. Dissipation of that proton gradient synthesizes ATP. Final electron acceptor in mitochondrial electron transport is oxygen. Those electrons being delivered to oxygen reduce it to water. So real quick, again, overview of what we already know about fuel oxidation. But what I want to highlight here is what we're going to see in this chapter with carbon fixation is pretty much an exact reversal of this. We take water and carbon dioxide, the two waste products of fuel metabolism, couple them with the energy from ATP, and we use that to synthesize carbohydrates and oxygen. So one of the things that we're going to see here is another piece that's important to that is where those sources of electrons come from. They do come from NADPH, but they actually originate from water here. So again, we're going to see some chemistry in our photosynthesis chapter here. We're going to see light and dark reactions. And so in the light reactions is where we see the oxidation of water. Again, those electrons are delivered to the chloroplast electron transport chain. Just like in mitochondria, the transit of these electrons through the electron transport chain generates a proton gradient, which is dissipated to make ATP. Just to highlight some differences here, we call that process photophosphorylation rather than oxidative phosphorylation. Final electron acceptor is NADP+, where it was oxygen in electron transport. Reduction of NADP+ generates NADPH, and it's those NADPH electrons that are used to reduce carbon dioxide, ultimately to 3-carbon carbohydrate precursors. And so those are going to be what are used to generate our fuel. So again, we generate oxygen because the oxidation of water generates oxygen. And so things that we have shown here in blue arrows represent what we refer to as dark reactions, and what we have here with black arrows represents what we see in light reactions. So it's very important to highlight, and I'll mention this several times throughout this review here. Light and dark reactions, even though we have the word dark here, oftentimes people will rename this light independent reactions because we want to highlight that these reactions actually are coupled processes that really both only occur during the day when there's light that's available. Dark reactions do not occur while it's dark out, but they don't require light. And so that's often why they're called light-independent reactions rather than dark reactions. So to highlight here some important big picture concepts, the light reactions are what we see with the electron transport chain in chloroplasts, the generation of ATP by dissipation of a proton gradient, and the production of NADPH from the final electron acceptor in the electron transport chain. The dark reactions, their purpose is to use that ATP and NADPH that's produced in the light reactions, use it to fix carbon dioxide or glue those single carbon atoms together to make carbohydrates. Specifically, the dark reactions are going to make 3-carbon carbohydrates. And then when we think about the chemistry that we know for reversing glycolysis, for example, pyruvate is a 3-carbon piece. Reversing that chemistry can allow us to make a lot of the carbohydrate precursors that we're going to see plants using to synthesize the carbohydrates that they need. So some really unique, just little stories that we talked about. Early discoveries of what was happening in this process. We knew that if we were to burn a candle underneath a beaker, for example, at some point the oxygen would be used up. And when the oxygen is used up, the wax as a fuel in the candle can no longer be combusted, and so the light goes out. However, if a plant were to be introduced into this system where there was no longer any oxygen, after a period of time, you could relight the candle, indicating that the plant was doing some chemistry that generated oxygen. So allowed sort of for a realization that these two processes could be related, right? We can sort of have a sustainable process where the oxygen that is liberated by the plant could be used to fuel the fuel oxidation that happens with the burning of a candle. So just kind of a neat little summary of some processes that we've studied thus far and we're going to elaborate on in this learning unit. So again, just another slide here that summarizes what we've talked about very generally there with light and dark reactions. So if we think about what happens in light reactions, and so here's an equation that you need to know. This was homework questions 2 and 3. The equation that we can think about for the light reactions is taking light energy to take the electrons that are lost from water. So again, the oxidation of water is that loss of electrons, but those electrons are fairly low in energy. So we need this light energy to excite those electrons to a higher energy state so that they can go through the electron transport chain. The excess energy can be used to produce a proton gradient. Again, photophosphorylation used to synthesize ATP. Final electron acceptor in the electron transport chain is NADP+ to generate NADPH. So again, another quick summary of the chemistry that happens in the light reactions. So a neat little just infographic here that kind of shows us that we have water coming in to supply those electrons, ATP and NADPH being produced by the light reactions. The oxidation of water generates molecular oxygen, and light is necessary for this process. We'll talk about this in a couple of slides here, but this has to do with the energetics of the electron transport chain. And when we have electrons that come in ultimately from water, they're relatively low in energy, and so they're not going to flow down an energy gradient in the electron transport chain unless we have some mechanism of making them higher in energy. And the light energy, photons of light, are going to be used to excite those electrons and make them higher in energy. Dark reactions are also referred to as the Calvin cycle. Again, I highlighted this is sometimes referred to as light-independent reactions. We use the energy from the light reactions, ATP and NADPH, that are produced to fix carbon dioxide and make carbohydrates. So again, here's the equation we want to think about and that I want you to know for the dark reactions. We've got NADPH, again, providing the source of electrons, ATP really providing some money or some currency for this process. So the electrons are given to carbon dioxide. Remember, electrons being glue that make bonds. We're able to glue these carbon atoms together to ultimately make carbohydrates. And then again, we liberate water here, and we liberate this water in the dark reactions ultimately from the polymerization reaction with carbohydrates. Now I want to make sure to highlight here, and we'll see this when we get to the details of the Calvin cycle, we really don't have individual carbon atoms from carbon dioxide that are zipped together one at a time. It's a very elaborate process that ultimately takes a 5-carbon piece, puts 1 carbon atom on to make a 6-carbon piece, and then does some unique sort of chemistry. Again, we had a really fun little video, a TED Ed video that we watched that kind of highlighted what goes on in the Calvin cycle. So again, a lot of comparing and contrasting that we see between these two chapters. And so we're talking about electron transport, and this electron transport happens between two different organelles that are both bacterial, essentially, in origin. So electron transport and oxidative phosphorylation from fuel metabolism occurs in mitochondria, and then electron transport and photophosphorylation occurring in chloroplasts in the photosynthesis processes. So what we're going to do here is just look at the anatomy of both of these, talk about what's similar and what's different. So just like we did, this is sort of a review from our electron transport chain. We're essentially going to have two membranes. We're going to have the outer mitochondrial membrane that we have on the outside here, which is highly permeable, and then the inner mitochondrial membrane, which is highly folded to create large amounts of surface area, and these invaginated or folded areas, these surfaces are called cristae. The space in between these two membranes is referred to as the intermembrane space. Outside of the mitochondria is the cytosol of the cell, and all the way in the interior of the mitochondria is the matrix. And typically this is very low pH when the mitochondria are not active, because we have not been pumping protons across these membranes via the electron transport process. So what we're going to see here is what we have in a homologous sense in chloroplasts. We have some structural differences. We still sort of have an invaginated inner membrane, but rather than kind of creating folds, we have discs that are created. So we have an outer membrane, which is comparable to an outer mitochondrial membrane, an inner membrane, which is comparable to an inner mitochondrial membrane. That inner mitochondrial membrane being folded and generating cristae in the mitochondria is folded to create thylakoid membranes or thylakoid compartments in chloroplasts. And rather than it being an inner membrane space, it's an inner membrane compartment. We still have the cytosol outside of the chloroplast and cytosol outside of the mitochondria, but inside of the mitochondria is referred to as matrix. The stroma is what is on the inside of chloroplasts. So again, protons are pumped in the same direction in the chloroplast from the stroma, which again, when they're not active, is going to have a relatively low pH. One main difference that I want to highlight, while both outer membranes are highly permeable, the inner mitochondrial membrane and the inner membrane in chloroplasts are both relatively impermeable. This is where most of the proteins for the electron transport chain are housed. But one very large difference is mitochondria are able to build up not only a concentration gradient, but a charge gradient. Okay, and this is very different from chloroplast, which is only able to build up a concentration gradient. It's unable to build up a charge gradient, and that's because this inner membrane, even though it's fairly impermeable to most things, is actually permeable to magnesium and chloride ions. And so by being permeable specifically to magnesium ions, as protons are pumped out, magnesium ions will flow in as a way to sort of counteract that efflux of a positive charge. And so we're going to see an influx of magnesium, and that neutralizes any charge gradient that could have been built up. So we really only see a buildup of a concentration gradient. So one of the things that I would like you to remember is that we do see kind of a difference in pH in mitochondria of about 0.75 units. And we actually see a difference in pH, and we'll talk about the details of this in just a little bit, in chloroplasts of about 2.5 to 3.5 pH units. So a much greater pH gradient is necessary because we don't get the extra thermodynamic driving force from a difference in charge. We don't have a charge gradient that's built up. We spent just a little bit of time talking about how plants actually harvest light. So we talked about why we see a lot of colors in plants. And these colors in plants are partly from chlorophylls, which we see here. They have two sort of regions that they absorb light. These are the main photosynthetic pigments that we have in plants. But we see a lot of other colored pigments that are responsible for harvesting light. Now, what's a really important thing to highlight is most of the chlorophyll that is actually in plants is actually not where we're seeing photosynthesis occur. It's just a mechanism for plants to be able to harvest and hang on to the light. So most pigments, most chlorophylls that we're going to see, or most other pigments like the carotenoid pigments and so forth, they are going to gather photons of light as they come in, and their own electrons are going to be excited into an excited state. what can happen is we're actually going to see the transfer of that energy via what we call exciton transfer or resonance energy transfer to an adjacent chlorophyll. And so the energy is transferred and that chlorophyll's electron gets to be excited to an excited state. So all of these light green kind of circles here represent antenna pigment molecules, where when a photon comes in, it excites the electron in that chlorophyll, and then the energy of that excitement is passed on to the adjacent chlorophyll, whose electron then is excited to its excited state. The sort of harnessing of this energy is relatively efficient so that we really don't lose much energy in this process. Now, what happens when we get to a reaction center chlorophyll, which is highlighted here in dark green, is we see kind of a jump down in energy. So energetically, you can sort of see these as being wells. So when we jump down in energy here, we don't come back out because that would be back up in energy. So once we get to these reaction center chlorophylls, once those chlorophylls electrons are excited, then they participate in the electron transport chain. So everything that we're going to talk about in terms of the electron transport chain, in terms of photophosphorylation and all of that, only occurs in photosynthetic reaction centers. So the two chlorophylls that we're going to talk about actually have wavelengths of light where they absorb at either 680 or 700 nanometers. So remember, a lot about spectroscopy has to do with matching energies with wavelengths of light. So again, to match the energy difference here for these chlorophylls, it's going to correspond to a wavelength of light about 680 to 700 nanometers. Just a fun little story that we talked about. Chlorophylls are very valuable pigments for plants, and every year, if it is a tree or a plant that tends to lose its leaves, what will happen is it will resorb these green chlorophyll A and chlorophyll B pigments. It will resorb those and hang on to those for next year. And why leaves turn color is all of these accessory pigments, like these orange carotenoids or some of these red pigments, are what then are able to be visualized in the leaf. And so you see these actually coming through. It's not that the leaf really changes color, but by removing all of the green pigments, you're actually able to more vibrantly see the other pigments. So, and remember, we had these videos are posted on Blackboard, but we had that neat little 5-minute video that kind of showed in an animated sense what happens in the electron transport chain. And we had all of those little chlorophyll molecules that kind of shook and resonated when they were excited by a photon of light. So the last slide that we kind of had with our first lecture was, again, doing a comparison, thinking about the chemistry that happens with the electron transport chain in mitochondria. So one of the things we're going to do is a compare and a contrast between where the electrons come from to start. So for mitochondrial electron transport, the electrons begin with NADH delivering its electrons on the matrix side. And NADH is a fairly high energy molecule in terms of its standard reduction potential. So from an energetic standpoint, it starts fairly uphill from water, which is the final electron acceptor in the electron transport chain, or I should say oxygen being reduced to generate water. But if we sort of walk through and review the pathway of electrons, electrons start again from NADH being delivered on the matrix side to complex 1. So again, NADH is oxidized to NAD+, and that's an important piece because we can then take that NAD+ and return it to get more electrons from the citric acid cycle, from glycolysis, from other processes that are going to generate NADH. From complex 1, those electrons are delivered to Q. Q also, again, is where complex 2 will deliver its electrons. And I didn't highlight it on this flow that we have here, but complexes 1, 3, and 4, as electrons pass through the electron transport chain, are responsible for translocating protons to generate our proton gradient. So every time we're able to go downhill in energy here, we can take that excess energy and use it to translocate protons and generate a proton gradient. So once we have our electrons at Q, going from Q to complex 3, again, that's an important process where we pump protons across through what we call the Q cycle. Then we deliver them to our hydrophilic mobile carrier, cytochrome C. Remember, Q is our hydrophobic mobile carrier. Take those electrons to cytochrome C, finally delivering them to complex 4, which is where molecular oxygen is going to be the final electron acceptor, and we're going to reduce molecular oxygen to generate water. So thinking about now what happens in chloroplast electron transport. And I highlighted to you guys and apologized. This is upside down here because I wanted to keep everything in the same orientation as we saw here for electron transport. But for some reason, the picture in your book shows it the other way, which again, I can think is a little bit confusing if we're always pointing as protons being pumped in this direction to go from the inside of the mitochondria out to the inner membrane space. I like showing the same orientation here to think about protons going from the stroma out into the thylakoid lumen, or what's referred to as the inner membrane compartment. So let's kind of highlight what's happening here. And we always kind of refer this a little bit in terms of not starting right at molecular water here, but if we start with chlorophyll that's in photosystem 1, photosystem 2 here, this is P680 chlorophyll. So what happens in photosystem 2 is we're going to oxidize P680. This chlorophyll is going to lose its electron and become P680 star. So just like we had NADH lose its electrons and be oxidized, this chlorophyll molecule is going to lose its electron and be oxidized and become P680 cation. So this P680 loses its electron. That electron gets excited, and we'd call this actually P680 star. So it's an excited electron. And then once it loses its electron, it becomes P680 plus. So now we've got photosystem 2. It's going to excite this electron. We're going to increase in energy from photons of light coming in. That excited electron is going to be passed down to Q. Now, we've got a cationic chlorophyll species that needs to have its electron replaced. So that also happens within photosystem 2. We've got what's called the oxygen evolving center. That's what's up here, OEC. The oxygen evolving center is going to take water. Water becomes oxidized to molecular oxygen, and then the electrons from that oxidized water are what are returned to that oxidized chlorophyll, returning it back to its regular P680 state. Okay, so that's the process that happens. Again, those electrons that are excited in photosystem 2 go on to Q, mobile carrier, that's analogous and homologous to what we see in mitochondrial electron transport. Then we deliver our electrons to cytochrome B6F, which is homologous to complex 3. Then they're delivered to a mobile carrier, plastocyanin, again analogous to cytochrome C. And then they're finally delivered to photosystem 1. In photosystem 1, we see another photon of light that's going to excite those electrons. They're delivered to an iron sulfur center that's going to be in ferredoxin. And ultimately, at the end of the day, we're going to deliver them to NADP plus to generate NADPH. So when we started with our kind of energetics of electron transport in mitochondria, it was basically just a straight downhill run because the electrons started as high energy because of their chemical composition as NADH. When we start with electrons in the chloroplast electron transport chain, those electrons are actually not high energy. Remember, they do originate initially from the chlorophyll, but they're replaced by water. So really the starting point for where those electrons come from is water. Water, again, is low in energy. So what we need to do is have those electrons be excited. And so we have wavelengths of light that are going to excite those electrons. Those photons excite those electrons. It happens in photosystem 2. And then we can kind of let those electrons go through an electron transport chain, use that energy to pump a proton gradient. Once we get to photosystem 1, we have to excite those electrons again. Exciting those electrons again allows us to go downhill once more, use that energy to generate a proton gradient, and ultimately the final electron acceptor in NADP plus is going to generate NADPH once it captures that electron. So a couple of things to highlight, just a funny little story. I don't think we talked about it in lecture, but this is called photosystem 2 and photosystem 1, not because somebody mislabeled them, but because this was the first photosystem that was identified and studied. And it wasn't until they found photosystem 2 and realized that it actually occurred earlier in the electron transport chain that they realized, you know what, we've got these numbered this way. We just start with photosystem 1. So just kind of an interesting little historical fact there. We could sort of call this electron transport chain energetics in mitochondria just a downhill slide. But what we call it in chloroplast is what we call a Z scheme. Now, I don't know why they don't call it an N scheme, maybe because they wanted to indicate Z for zigzag. But every time we need to excite our electrons to a higher energy state, we use photons of light, again, relaxing down to allow us to generate a proton gradient, and then we excite one final time here to finally get to our final electron acceptor. So this is called a Z scheme because of the change in the standard reduction potentials as we excite our electrons and then allow them to go through the electron transport chain. So our first lecture really was kind of an overview of the process, doing a lot to review and compare and contrast what we saw in Learning Unit 4 on electron transport and oxidative phosphorylation in mitochondria. So again, seeing a lot of homology with mitochondria, so a great way to sort of review what we have there. So in our next lecture, we sort of moved on and took a deep dive on what these complexes are in the electron transport chain and then for photophosphorylation. So again, we started out with just a review again of the energetics of electron transport, both in mitochondria here, where it's basically just a straight shot down from NADH, which is high energy by virtue of its chemical composition, and then the electrons in chloroplast electron transport are high energy by virtue of being excited by photons of light. So again, here we can just see this Z scheme here. Both of these, again, showing standard reduction potentials as energy, and we can see here as we go through photosystem 2, down to Q and the Q cycle in cytochrome B6F, and then in photosystem 1 being excited again to finish out the process, delivering those electrons to NADP+ to generate NADPH. So a little bit here now to go through the details of what we see with each of the complexes. So just to highlight, we are going to be just kind of taking this picture that we have here and talking about these complexes one at a time. So I'm just going to run through them real quickly here on this little picture so that we can see. We're going to start first with photosystem 2, and then we're going to talk about Q, the mobile carrier that lives in the inner membrane of the chloroplast. And then we've got cytochrome B6F that we have there. We've got photosystem 2. Those electrons are delivered by plastocyanin. And then we've got ferredoxin that we have here, ultimately delivering to what we call FNR. And that's where we finally deliver those electrons to NADP+. So again, looking at these complexes one at a time, you have a homework question that kind of sums these up. So what we're going to see here is we're going to highlight where the electrons come from, where they go to, and how we translocate protons if it's applicable. So again, starting with photosystem 1, the electrons originate from P680 chlorophyll, and they ultimately go to our mobile carrier Q. And so we really don't have any translocation of protons here, but I just wanted to highlight here that when that electron is excited and then leaves, so we excite that electron and we get what's called P680 star, and then when we lose that electron, we get P680 plus. So again, P680 is the chlorophyll here. It's going to lose its electron to go through kind of the electron transport chain through some redox centers here in photosystem 2. We are going to replace that electron from water that is oxidized in the oxygen evolving center. So that's what our next piece is here. It's actually part of photosystem 2. So the oxygen evolving center that's in photosystem 2 takes electrons that originate in water and ultimately give them to P680 plus, regenerating it in its P680 kind of form. The chemistry for what happens here is, again, water is oxidized in the oxygen evolving center, and these 4 protons here are, since this process happens on the inner membrane compartment side, it's kind of the equivalent of putting those across the inner membrane there, and so that helps to contribute to the proton gradient. Oxygen is evolved. We know that happens with chloroplast electron transport here. And then again, those electrons are given to that P680 cation here to regenerate that P680 chlorophyll in its uncharged state. Okay, so kind of highlighting that again here, we're going to have P680 lose its electron, ultimately going to Q. The electron in that chlorophyll is replaced by the oxidation of water to replace that electron. And then we have protons that are contributed to the intermembrane compartment, and that helps with the proton gradient. So we're going to see three spaces along the this journey here where we contribute to the proton gradient. Really only one of them is truly proton translocation, and we'll see that in our next kind of sequence of steps here. So again, once we have the electrons at Q, they're going to be coming from photosystem 2, and then they're going to be going to cytochrome B6F, which is kind of the homolog to complex 3 in mitochondria, which is the same direction that we saw. Q delivered electrons to complex 3 in mitochondria, and in doing that delivery, that was the Q cycle. We didn't go into the details of Q cycle in mitochondrial electron transport, but that's a very important process to transport protons to the intermembrane compartment. So again, homologous Q that we have in mitochondria, we see in chloroplast here. So now the cytochrome B6F is homologous to complex 3. So it has brought electrons in from the Q cycle because they come from Q, and they're ultimately going to be going to plastocyanin, and plastocyanin again is the homolog to cytochrome C. So this is a very important process for sort of translocating protons. Just like we talked about the Q cycle in mitochondria, we double our thermodynamic efficiency by moving electrons through the Q cycle. So again, we're going to have these protons here that are dumped over to the intermembrane compartment. So electrons, as they leave cytochrome B6F, are delivered to our hydrophilic mobile carrier, plastocyanin. So plastocyanin, again, being homologous to what we saw with cytochrome C. So again, getting its electrons from cytochrome B6F, delivering them to photosystem 1. No proton translocation is really happening here. And similarly, there's really no proton translocation that's happening in the next step. And that's where photosystem 1 is ultimately, again, getting those electrons from plastocyanin and then delivering them to ferredoxin. So this is, again, another place where we have photons of light coming in. So these electrons that come in from plastocyanin are relatively low in energy. They get excited in photosystem 1 and delivered as higher energy electrons to ferredoxin. So again, we've got these ferredoxins. If we refer to this as FES on a previous slide, that's just because there's an iron sulfur center that we see in ferredoxin. But again, no proton translocation at this point. So to highlight the three places that we see contributions to the proton gradient, are from the oxygen evolving center, the protons that are released from water, because they're delivered to the intermembrane compartment, we're going to see them contribute to the proton gradient. We've got the Q cycle that has sort of a direct translocation of protons into the intermembrane compartment. And the last thing that we're going to see is really, again, not a movement of protons, but when we have ferredoxin deliver its electrons to FNR, and so FNR is ferredoxin NADP plus reductase. So this is an enzyme where ferredoxin docks in, delivers its electrons. NADP plus is sitting in the enzyme active site, ready to take those electrons and generate NADPH. Because the protons to complete this process are actually taken from the stroma, it's equivalent of contributing to the proton gradient in that sense. And so again, we're generating NADPH from NADP plus being our final electron acceptor in this electron transport chain. So finally, looking at this process again, right side up now, boxed in red here, we've got the three spaces that we're going to contribute to the proton gradient. Again, the Q cycle contributes directly by moving protons into the intermembrane compartment or the thylakoid lumen. We've got protons that are generated on this intermembrane compartment side by the oxidation of water. And then we've got the removal of protons to generate NADPH on the stroma side. So all three of those contribute to the generation of a proton gradient. Just highlighting that the two main products that we see in these light reactions, which are chloroplast electron transport, are NADPH. And we're going to talk about on the next slide, synthesizing ATP. We're not going to spend a lot of time talking about this because if there ever was a near perfect homolog to what we see between mitochondria and chloroplasts, it's what we see here with this proton translocating ATP synthase. So this is again homologous to complex 5 to ATP synthase that we see in mitochondria. We're going to deliver those protons back from the inner membrane compartment back to the stroma, same type of a lollipop head, merry-go-round binding change mechanism that we saw in mitochondria. We see the exact same process happening in chloroplast electron transport to synthesize ATP. So one other thing to highlight that makes chloroplast electron transport a little bit unique is that once we get to this spot with ferredoxin, again highlighting right here, once we've gone through this electron transport chain to ferredoxin, we don't have an arrow here. These blue lines represent the flow of electrons. What we have shown here is the non-cyclic pathway to electrons. But something that can happen in some plants is we have the ability for ferredoxin not to deliver its electrons to the final electron acceptor, but to actually say, you know what, I'm going to sneak back here, I'm going to ride the ride again, and I'm going to give my electrons to the Q cycle, and in doing so, generates an even greater proton gradient and contributes more to the synthesis of ATP than to the generation of NADPH. So highlighting that here, again, ferredoxin, we have a bifurcation in this pathway where we have two options. Ferredoxin in that non-cyclic way can take its electrons and then deliver them in FNR to NADP+, generating NADPH. Or in a cyclic pathway, ferredoxin can deliver those electrons over to Q. Q becomes reduced again, and in pulling two protons from the stroma and then ultimately delivering them over to the inner membrane compartment, or that thylakoid lumen, we generate a greater proton gradient and we increase the production of ATP. So again, to highlight, the cyclic pathway will have an elevated amount of ATP synthesis. Eventually, those electrons will make their way to their final electron acceptor. So we're still going to make NADPH, but by using this cyclic pathway, we have the ability to sort of bump up the amount of ATP that we can make. So just giving a little preview here, we're going to see a couple of options of where ferredoxin can take its electrons. So we see two right now, can take its electrons to NADP+, it can take its electrons to Q, and just as a preview, we're going to see this when we talk about regulation, ferredoxin can take its electrons and deliver them to a redox cascade that ultimately is going to reduce two enzymes that are important for regulating flux in the dark reactions in the Calvin cycle. So we'll see that in just a little bit. Not too much that we're going to talk about here with ATP synthase. Remember ATP synthase, we actually called an F1, F0 ATPase in mitochondria. We call this one a CF1, CF0 because C just refers to the fact that it's a chloroplast ATPase. Homologous to what we see, the same type of a mechanism, we've got that binding change mechanism. But one of the things that I wanted to highlight, and I mentioned these numbers early on here, is the delta pH, the difference between the pH on the inside and the outside of the inner mitochondrial membrane is about 3/4 of a pH unit. We see a much greater pH difference across the inner membrane in chloroplast, and that's because of the permeability of that membrane to magnesium and chloride. And it's really the magnesium piece to it that's more important, because as protons are pumped out, we're going to see magnesium is allowed to flow in, and that's going to negate any potential charge gradient that was built up. So remember in our electron transport and oxidative phosphorylation learning unit, we saw this equation that just reminded us that the thermodynamic driving force for the generation of the proton gradient really had two parts. It had the fact that we are physically building up a chemical gradient between a chemical species, protons, but because those protons were also generating a charge gradient, we actually had an additional contribution to this thermodynamic driving force. Now, we highlighted again that that is not going to be possible when we have chloroplast electron transport because of the permeability of that inner membrane to magnesium. So again, that summed up our second lecture that we had there. And then we moved on to discuss regulation and then importantly, what happens in the dark reactions. So again, kind of summarizing what happens in the light reactions, we're going to have chlorophylls that are capturing photons, harvesting that energy, but it's not until that energy gets to a reaction center chlorophyll that we see the light and then the dark reactions that we're going to talk about in this lecture here. So electron transport in chloroplasts, we're going to use, again, the splitting of water to ultimately generate those electrons that are excited in two different photosystems that have chlorophylls of different wavelengths of light that they're going to absorb. We generate a proton gradient. Again, we generate that by not only splitting water, by pulling protons to generate NADPH, but importantly through that Q cycle. That Q cycle is important because when we have that cyclic electron path, we can actually increase that concentration gradient that we're going to make with protons. And so we're going to be able to make even more ATP. So ATP synthase is going to be responsible for synthesizing ATP, and we're going to dissipate the proton gradient that we have to do so. And then ultimately we're going to synthesize NADPH because we're going to use those electrons and deliver them to NADP+ to generate that NADPH. So that's the light reactions. So talking about the dark reactions ultimately is using that NADPH and ATP to fix carbon dioxide. So why are we again reviewing something else that we already talked about? Well, that's because the pentose phosphate pathway and the Calvin cycle have a lot of homology. Oftentimes the Calvin cycle is referred to as reductive pentose phosphate pathway. So if we highlight kind of what's going on here, and I'm going to draw a lot of attention to the first stage of the pentose phosphate pathway. And again, don't worry about the chemistries that we see happening here. Don't worry about those equations. Our homework questions really asked us to think about the three main stages that we see in the pentose phosphate pathway, or the two main stages that we see in the Calvin cycle. And again, we're highlighting on this slide here, what are the important things that I want us to remember? Well, first stage of the pentose phosphate pathway is important because that's where we generate NADPH. We liberate carbon dioxide, and the enzyme I want you to remember for that is the rate determining enzyme, which is G6PD. The end point of stage 1 of the pentose phosphate pathway is to generate ribulose 5-phosphate, and that's important because that's a common piece that we see here in the Calvin cycle in the dark reactions. So again, stage 1 of the pentose phosphate pathway is synthesizing NADPH, liberating carbon dioxide, and again, the rate determining step for that process is G6PD. The purpose of the second stage of the pentose phosphate pathway, remember, is to determine how we're going to restructure ribulose 5-phosphate. How many ribose 5-phosphates and how many xylulose 5-phosphates are we going to make? Ultimately, because if we want to make a lot of nucleotides, we're going to make a lot of ribose 5-phosphate. If we need to just recycle these carbon building blocks, we're going to use the third stage here, and we're going to recycle them into 6-carbon and 3-carbon glycolytic intermediates to be able to fully be recycling those products in glycolysis. So what we're going to see here is there's 2 stages to the Calvin cycle that have some homology to what we see with the pentose phosphate pathway. Stage 1 is essentially kind of a reversal of what we saw in stage 1 of the pentose phosphate pathway. This is where we're going to actually be producing GAP by fixing carbon dioxide. So we're going to be using NADPH, not generating it. We're going to be using carbon dioxide, not liberating it. And because this is going to be an uphill process, we're going to need to add a little bit of energy in. But at the end of the day, we generate GAP, and that's important because we can siphon this off to use to synthesize carbohydrates. Now, as we learn the details for this process, we'll actually see that we're going to make 6 molecules of GAP, but we only siphon 1 of them off for carbohydrate use. Other 5 need to be restructured in a recovery phase where we're going to take 5 GAPs, so 5 C3 pieces, and we're going to reshuffle them to make 3 C5 pieces, and some really interesting reshuffling, if you will, and we watched a neat little video on that to highlight how that process happens. So again, just looking at some of the structures, not anything that you need to know, but kind of wanted to box on this diagram since this was a diagram we talked about in the pentose phosphate pathway. Again, important pieces is that we generate NADPH and liberate CO2 in the pentose phosphate pathway. Rate determining step, G6PD. Endpoint of stage 1 is ribulose 5-phosphate. And again, we have to generate at the end, if we want to recycle our intermediates, GAP and F6P. In the Calvin cycle, and we kind of didn't mention this name, RuBisCO is the important enzyme. It is one of the rate controlling enzymes in this process. And as we talked about in class, it arguably could be the most important enzyme in the world, because if plants did not have the ability to fix carbon dioxide as fuels, we would have nothing to eat. So again, ATP is necessary for energy. NADPH is necessary for electrons. These are what are generated in the light reactions. And so the purpose of the light reactions are to fix carbon dioxide as 3 carbon pieces. And I mentioned this early on, but we want to highlight this is not saying that we take individual single carbon pieces as carbon dioxide and put 3 of them together to make GAP. Rather, we take a 5 carbon piece, attach 1 carbon on to make a 6 carbon piece that gets split apart into a 3 and a 3. And then we've got these 3 carbon pieces that we're going to siphon 1 off and then do some recycling, some restructuring to regenerate our 5 carbon pieces. So some really neat chemistry that sort of happens in this second stage here that we're not going to get into the details of. I am going to just spend a brief moment here talking about the chemistry that happens with RuBisCO. We're not going to look at this mechanistically, but I do want to highlight that what we have here is we have a nucleophilic species that we generate by deprotonating ribulose bisphosphate, and that nucleophile is going to react with CO2 as an electrophile. So why it's important to just kind of highlight that here is what we're going to see in a couple of slides is in certain situations when carbon dioxide has been depleted from sort of the local atmosphere, RuBisCO still has to function, and we'll talk about why that is. But what's going to happen then is it's going to use oxygen in its place. So again, this is going to be some unique chemistry that can happen with RuBisCO. We're going to talk about when and why it can happen in just a few slides here. So to summarize the dark reactions, we're going to basically use the ATP and the NADPH that we made in the light reactions. We're going to use that to fix the carbon from carbon dioxide into a usable form as 3 carbon GAPs, which are important to synthesize other carbohydrates. So at the end of the day, we're going to use some other Calvin cycle enzymes to ultimately get to F6P. And we sort of talked about what those enzymes were. And so, again, one of your homework questions was to think about how we could run glycolysis or enzymes of glycolysis in reverse, thinking about gluconeogenesis, to take GAP and make it into F6P. So we'd have TIM that would take us from GAP to DHAP. Reversing aldolase will take us from DHAP and GAP into FBP. And then FBPase, again, the rate controlling step of gluconeogenesis, will take us back to F6P. Once we're back at F6P, some unique chemistry will allow us to not only epimerize, but do mutase chemistry to move the phosphate from the 6 position to the 1 position and convert from a ketose sugar to an aldose sugar to get to G1P. And once we have G1P, now we have the ability of making a bunch of different carbohydrates depending on how we activate that monomer to build. So a couple of slides here, and we had a homework question here with a nice chart that allowed us to sort of summarize the different pieces. You don't need to, remember specifically which one is which, but I do want you to know that depending on what nucleotide we use to decorate our G1P, because remember, we're going to basically take our glucose building block and add on a good leaving group. So depending on the identity of the nucleotide that's on that leaving group, kind of tags these building blocks for different things within plants. So if we want to make starch, again, I do want you to know that building starch is going to be making alpha-1,4 glycosidic bonds. Starch is like making glycogen in you. This is how plants store energy for kind of a long-term or use at an alternative time. And so starch synthase is going to be the enzyme that synthesizes those alpha-1,4 glycosidic bonds. And it's going to know that we have its substrate because we use adenosine as an ADP good leaving group. Thinking about 2 other sort of carbohydrates that plants are going to make, and these are differentiated too by where they're made. So starch synthesis occurs in the chloroplast, whereas sucrose and cellulose synthesis occurs in the cytosol. Sucrose, again, being unique is that we're going to have an alpha beta 1,2 glycosidic bond that's synthesized. So that head-to-head linkage that we're going to have with sucrose synthesis, making that non-reducing sugar. How we're going to know that glucose is being tagged for that process is we're going to see UDP glucose for that. We're also going to see UDP glucose, again, in the cytosol being used as a substrate to synthesize cellulose, again, having that beta 1,4 glycosidic linkage. So just to highlight what these are useful for, sucrose is important as kind of a water-soluble, transportable fuel. So plants have a vascular system, and so how they're going to move fuel around to non-photosynthesizing cells, like you might find in the roots, for example, is they're going to have sucrose that they're going to be able to transport. Starch, being a large carbohydrate polymer, is not water soluble, and so it's not going to be able to be transported as easily as sucrose. Again, cellulose, really important. It's the main structural component that we're going to see with plants. So the last thing that we spend a little bit of time talking about is the importance of differentiating the metabolic processes that happen for plants, whether it's daytime out or nighttime out. So kind of thinking about how we can easily sort of differentiate that, it really depends on what organelle is doing chemistry. And again, it's not to say that this is completely binary, that there's no mitochondrial function during the day, but by and large, we can think about this as chloroplasts being active during the day and mitochondria being active at night. So again, chloroplasts are going to harvest light energy, and that light energy via the light reactions is going to synthesize ATP and NADPH. The ATP can not only be used for cellular needs, but it can be used with that NADPH in the dark reactions to synthesize carbohydrates by fixing carbon dioxide. So again, during the day, chemistry is going to be monopolized by the chloroplast as an electron transport organelle. We're going to have the electron transport chain synthesize ATP and NADPH, and then assimilate and fix carbon dioxide in the Calvin cycle or the dark reactions to make carbohydrates. So what do plants do at night? Well, because this process isn't available, if they want to synthesize ATP and NADPH, they have to use their mitochondria and they have to do the same metabolic processes that we've already learned about. So they'll take carbohydrates in the pentose phosphate pathway to make NADPH. They'll take carbohydrates and break them down via glycolysis, the citric acid cycle, electron transport, and oxidative phosphorylation in mitochondria to synthesize ATP and again, waste products of CO2 and H2O. Now, what's really important to highlight is if we were making any ATP and NADPH and the dark reaction chemistry was still going on at nighttime, we would essentially be breaking down carbohydrates to make ATP and NADPH, and then using ATP and NADPH to fix carbon dioxide and make more carbohydrates. That would be a futile cycle. So it's very important that RuBisCO be inhibited at night. We do not want the dark reactions happening at night. So how do we think about doing that? Well, again, the dark reactions being inhibited at night. Let me just go back here and highlight for a second. It's easy to inhibit the light reactions at night because there's no light. So we're not going to have photons of light coming in, so no electron transport is going to be happening. However, the dark reactions being light independent, unless we have some mechanism for inhibiting them, we're not going to be able to stop those processes from happening. So just like we've done before, to identify flux control points, we look for reaction chemistries that have large negative delta Gs. They're going to represent good control points. So we're going to see RuBisCO here as an enzyme that we'll talk about. And we'll also see two enzymes that are important for this recovery phase in the Calvin cycle, FBPase and SBPase. And here, just kind of an equation form that we talked about in question 15, just another way to think about the chemistry that happens in the dark reactions, and then the chemistry that would happen, and again, not in the dark reactions, but actually physically in the dark if it's nighttime out. And again, we don't want this futile cycle where we're taking the NADPH and ATP that's generated by catabolic processes and using it for the anabolic process of the Calvin cycle. So we're first going to talk about RuBisCO here. And so there's three ways that we talked about controlling RuBisCO. Two of them have a direct sort of connection with what's going on with light reactions. So if we think about it, if there's light out, what's going to be happening is lots of electron transport. If there's lots of electron transport, we're actually going to be increasing the pH of the stroma. Remember, we're moving protons out of the stroma. So that means the concentration of protons is lower, which means the pH is higher. So RuBisCO just naturally operates best near in a pH near 8. So as that stromal pH increases, as we're doing proton pumping, that's going to be happening when there's light out. We're going to make we're going to make RuBisCO more active. Similarly, if we're doing a lot of electron transport, that means we're going to be pumping protons out, and magnesium is going to be flowing in. So it's not surprising then that we have RuBisCO being stimulated by magnesium. So two signals that are strong indications that electron transport is going on, which must mean it's light out, is an increase in pH in the stroma and an increase in magnesium concentrations. Both of those, again, are going to be signals to activate RuBisCO. Well, that is all well and good. At nighttime, again, the pH is going to fall, magnesium concentrations are going to fall, but it's a fail-safe to make sure that we have RuBisCO not active at night. There's going to be a transition state analog that is actually synthesized at night that's going to help inhibit RuBisCO. And this should say RuBisCO, not RuBP. It should be either RuBP carboxylase or saying RuBisCO. So inhibiting this enzyme happens at night, not just because we have the pH drop and magnesium concentration drop, but we also synthesize a transition state analog kind of as a fail-safe mechanism to make sure that that stays inhibited. All right, last little piece to talk about here in terms of regulation was thinking about a redox cascade. So use this as a mechanism to kind of review phosphorylation cascades. So again, homework question 18 sort of talked about the phosphorylation cascade that starts with a low blood sugar signal. So glucagon. Glucagon is going to activate adenylate cyclase. Activating adenylate cyclase increases cyclic AMP levels. Increasing cyclic AMP levels causes that R2C2 inactive tetramer to dissociate into the catalytic subunits, which can phosphorylate many things, but in this cascade that we learned about, it was phosphorylating the alpha and the beta subunits of phosphorylase kinase. Coupled with the binding of calcium to the delta subunit, the catalytic or the gamma subunit in this kinase underwent a conformational change, removed its own inhibitory domain, and then could then phosphorylate its downstream substrate, which was the serine 14 on phosphorylase. Now that serine 14 is phosphorylated, remember we can't bury it. By not burying it, we also have it be locked into place on the outside with those arginine residues. Phosphorylase is active and we break down glycogen. That's going to remedy a low blood sugar level. Okay, that was a lot. That was a review, but again, just reviewing what happens in a phosphorylation cascade. Now we're going to think about what happens with a redox cascade, which is really an electron cascade here. So remember, we talked about there being the potential for another option for something to happen with ferredoxin. So I'm going to flip back here just for a second to remind us of where ferredoxin is in this process. So again, ferredoxin is right here. So remember, we had the potential for ferredoxin if we talked about a cyclic pathway of electrons. So ferredoxin in a non-cyclic pathway could take its electrons over to FNR. In a cyclic process, it could return them to Q. We're going to talk about the third pathway for things that can happen here. And the third option, my screen won't move here, there we go. My third option for what we can do with electrons here is to deliver them to this redox cascade. So this was homework question 19 here. So again, we just talked about ferredoxin delivering them via the non-cyclic pathway to NADP+, the cyclic pathway delivering them to Q, and it can deliver them to this ferredoxin thioredoxin reductase cascade. So again, here is ferredoxin, here is the starting point. So when ferredoxin has its electrons, it can deliver its electrons to ferredoxin thioredoxin that's in an oxidized state, so then that becomes reduced. This guy then can deliver its electrons to thioredoxin starting out in an oxidized state, so now it's reduced. Thioredoxin delivering its electrons to bisphosphatase enzymes that are in an inactive state. Being reduced puts them in an active state. So these bisphosphatase enzymes are these two guys right here. These enzymes are active when they are in a reduced form. We haven't really talked about this too much, but again, lots of things can cause enzymes to undergo often conformational changes that allows them to become active. In this case, we've got two cysteine residues that when they're hooked together, this protein is going to be in a conformation that is not suitable to be active. However, when we reduce and free up those two cysteine residues, adopt a conformation that's now able to do chemistry with its substrate. So an example of a redox cascade here. So just kind of summarizing here, another homework question that just really got us to think about, well, what's happening when it's light out? What's happening with it when it's dark out? And adding in one more little piece here. So again, when it's light out, we're going to be doing the light reactions because there's photons. We're going to be having electron transport happening. We're going to be then increasing the pH of the stroma, increasing the magnesium concentration in the stroma. So that also means we're going to be activating the dark reactions. The dark reactions being the Calvin cycle. So again, we've just talked about how we are going to be activating RuBisCO by having an increase in pH in the stroma and an increase in magnesium concentration in the stroma. But we just learned about how an increase in the electron transport chain can have ferredoxin deliver some of its electrons to that ferredoxin thioredoxin redox cascade, ultimately reducing FBPase and SBPase so that these guys are active, again facilitating chemistry in the Calvin cycle. One of the things that we didn't talk about, and we didn't talk about the mechanism for how this happens, but when we're having this process happen, we are going to be inhibiting PFK. Remember, PFK is the rate determining enzyme of glycolysis. This is really important. If we have the ability to go through processes that not only generate ATP and NADPH, but generate carbohydrates, we do not want to be breaking down fuels to generate ATP and NADPH. So we need to be inhibiting fuel breakdown, and we do so by inhibiting PFK. Flip the lights off, right? When it's dark out, there's no photons. If there's no photons, electron transport chain is inhibited. If there's no electron transport chain, we're going to be dropping the pH in the stroma, dropping the concentration of magnesium, and we're also going to be synthesizing that transition state analog. All of these are going to be inhibiting RuBisCO. We do not want the dark reactions. We do not want the Calvin cycle operational at night. We also then relieve the inhibition and thus activate PFK, and we're going to be allowing for fuel breakdown, and that is how plants will generate the ATP and the NADPH that they need at night to do their biochemical functions. The last little piece to talk about here is this concept of photorespiration. Remember early on I talked about the enzyme RuBisCO, and I said the normal substrate for RuBisCO is going to be carbon dioxide. We're going to be deprotonating this RuBP here, generating a nucleophile that will attack our CO2 as our electrophile. Well, what happens is it's possible if there's not sufficient CO2 around and RuBisCO is hyperactive, and we'll talk about why and when that would be the case, it needs to do its chemistry. So it's going to grab an alternative substrate and it's going to grab oxygen. It's actually going to end up generating peroxides, and we're not going to talk at all about the chemistry that happens. I just want you to know that RuBisCO can use oxygen in addition to its sort of native or natural substrate. When it uses oxygen, we call this process photorespiration. So by definition, we're going to be using oxygen and evolving carbon dioxide. That's why we actually call it respiration, because that's what we do when we undergo cellular respiration is we use oxygen and we liberate CO2. Again, this is not saying that it is this reaction that's happening that's doing that, but that's why we call it respiration, because the net is the same. We're going to use oxygen and liberate CO2. All right, so why the heck would this be happening? Well, if there's not CO2 around as a substrate, but we've got lots of light out, that means we're doing all of these things to hyperactivate RuBisCO. If there's lots of light out, there's lots of electron transport, we've got a high stroma pH, high magnesium levels, RuBisCO is hyperactive. In order to kind of keep this process flowing and not back up electrons in the electron transport chain, we have to keep RuBisCO just doing its chemistry. So we give it just something to do chemistry with. We give it oxygen because there's actually going to be plenty of oxygen around if there's not CO2. And the main reason that we do this is if we were to kind of back up electron transport chain and we'd have all of these photons bombarding our chlorophyll with photons and exciting them and electrons all over the place, that actually will lead to photooxidative irreversible damage to chloroplasts, and that actually will kill a plant. And so they need to have some way of allowing electron transport to occur. We're not going to go through the details for this. One of the things to highlight is it actually it's a little bit of a futile cycle. It costs ATP to do this. But you know what? If you've got so much sunlight out that you've depleted your CO2 because you're just like ripping through these processes really rapidly, you can be a little bit wasteful because you're just like growing money on trees, if you will. So one of the ways that a plant sort of gets around having to do photorespiration, because it's not ideal to do that, is it finds a way to sort of stockpile carbon dioxide. And so these kind of plants are what we call C4 plants. So just to highlight here, question 22 on your homework asks you to think about what it means to be a C3 plant. And that's where RuBisCO is just kind of doing its normal chemistry. It's taking and it's fixing carbon dioxide and it's making C3 molecules. It's making the standard GAP. So that's kind of the standard quo or the status quo. What RuBisCO wants to do is make 3 carbon pieces. And so most plants that we see sort of in our climate are what we call C3 plants. These plants are not really equipped to think about doing a lot of photorespiration. They don't really need to, because if we think about plants that live, say, in Erie, there's not a lot of sunlight year round. It doesn't get overly hot. It doesn't get overly light. So we don't tend to have a situation where we're doing so much light and dark reactions that we deplete the carbon dioxide that's kind of in the local atmosphere. However, if you live where it's very sunny out, there's a good chance that you might be doing so much electron transport and light reactions, so much dark reactions and depleting carbon dioxide that you might actually run out in your local environment. So what plants will do there is they actually are called C4 plants because they will stockpile carbon dioxide as a 4 carbon molecule. You do not need to know the details of this, but I do want you to know that C4 plants operate kind of in one of 2 fashions. Kind of a standard C4 plant is they actually separate things in space. So you have 2 different kinds of cells here. You have mesophyll cells and then bundle sheath cells. So mesophyll cells do not have the biochemical machinery to actually do the Calvin cycle. So what they'll do is they'll take and they'll sequester carbon dioxide by fixing it to the 3-carbon phosphenylpyruvate, making it into the 4-carbon oxaloacetate, and ultimately doing some reduction chemistry to generate the 4-carbon malate. So malate, again, you can think of as being sort of a storage reservoir for carbon dioxide. If this plant happens to be doing a ton of Calvin cycle chemistry where they're beginning to run out of carbon dioxide in the atmosphere, it will actually pull malate from its mesophyll cells into these bundle sheath cells. They'll use an enzyme that will regenerate that CO2 and allow it to be used kind of in the Calvin cycle. So that's kind of some unique chemistry where we can stockpile and store carbon dioxide as malate in a different kind of cell and pull on it when we need it. So again, just as a highlight, we talked about the fact that a lot of weeds that we see in our local area tend to be C4 plants. Right? If you think about your grass looks beautiful in the springtime when it's just starting to grow. It's not overly hot, not a ton of sun, but as we get into like the June and July weather, we start getting really warmer. And once you get into August, if you've not sufficiently watered your grass so that your grass is able to kind of really grow really well and grow and crowd out the weeds, what can happen is that these weeds are very heat tolerant and they're very sun tolerant. And that's because they're C4 plants. So they're actually able to monopolize kind of space resources in your grass, and they're going to start taking over. So the last thing to talk about is, well, what if you're a C4 plant, but not only do you live where it's really hot, but you live where it's really dry? So one of the things that we didn't talk about the specifics of is how plants actually get these gases, and specifically water, into their tissues, if you will. Plants have what we call stomata, and they're literally like pores that are on their leaf surface. Now, the problem is if you live where it's very arid, what's going to be problematic is if you open up your pores during the day to try to get some carbon dioxide in, you're going to let water out and you're going to dry out. So plants that live in the desert cannot open their pores during the day to pull in the carbon dioxide they need. So they only open their pores at night. And so these plants actually will sequester carbon dioxide in time rather than space. So they do the same kinds of chemistry. They're going to take carbon dioxide, they're going to fix it to phosphenylpyruvate, and they're going to generate malate. But now these plants are just going to store it kind of in the mix. And then as they need it during the day, they're slowly going to kind of trickle it out. And so they never pull in carbon dioxide during the day because they can't open their pores. So really neat sort of system here where they sequester and separate carbon dioxide acquisition in time rather than space. So didn't really go over this slide too much in class, but just kind of highlighting the difference between C3 versus C4 plants. And again, most plants that we talk about fix carbon dioxide as GAP. C4 plants are going to store carbon dioxide as the 4-carbon malate. And so kind of talking about where you'd likely find these plants, kind of advantages to sort of having these different systems, and again, disadvantages. So disadvantages to sort of a C4 plant is they are not as energy efficient because they are sort of limited by some of the processes that they can do. But you know what? It doesn't matter if you're energy inefficient, if you literally grow money on trees by having so much light available to do that. So, end of photosynthesis. I hope you found it interesting, and I hope you said, holy cow, I did not think plants could be so interesting. And wow, that was an amazing way for me to review the content that I needed to know for mitochondria and electron transport and oxidative phosphorylation.